Hiya! Okay, so this is key area 4 of Unit 1 of National 5, and it is about proteins. Really, I think it's named wrong because it really should be called enzymes. That's mostly what it's about. But we'll start off with a little recap of the proteins that we looked at last time, and then look at enzymes in a lot of detail.
Okay, so again, from the SQA course specification, here's what you need to know throughout this key area. We're going to focus on the first two today. Yeah, first two in this video. Again, you can pause it and have a read of this if you like, but we're just going to get going.
So we'll be looking at the 5 main functions of proteins. We've already covered those, so we're just going to skirt over those. Enzymes being catalysts, enzymes and substrate interactions, and their optimum conditions as well over this entire key area. So it's going to be the first 3 during this video.
Okay, so just to recap those 5 main functions of proteins, we have structural, hormonal, antibodies, receptors, and enzymes. If you can't remember what these are, watch the last video.
Yep. Really? That is saying share down the side. Okay, and enzymes is bolded and underlined because that is the main focus of this entire key area. Yeah. It's also the thing that's going to be asked about the most in your exam paper. There's usually enzyme experiments, all sorts of enzyme graphs, tables, kinds of things that they can ask about. So it's a very popular area with the SQA.
Okay, so looking at enzymes now. So we're going to look at several key facts. We're going to look at the first 4 in this video and then the other 2 in the next video. So we're going to look at the idea of enzymes are made by all cells. They speed up reactions. They're specific and they can be involved in degradation or synthesis reaction. But all of these key facts you need to know about enzymes.
It's about, for this, it's about being able to explain that sentence. If you can explain that sentence in terms of your knowledge, you will be fine. Okay, so if you can get these 6 key facts sort of in your head and be able to explain by what do you mean they're made by all cells or what do you mean they're affected by pH, you should be okay.
Okay, so in this we're going to learn about the role enzymes play in speeding up chemical reactions in order to sustain life. So by the end of this you should be able to state what a biological catalyst means, state where enzymes are produced, state what enzymes do in a reaction, name the substance an enzyme acts on, and name the item made at the end of an enzyme reaction.
Okay, so to recap, enzymes are biological catalysts. We had a look at that in the previous video. A biological catalyst is something that speeds up cell reactions and it's produced by living cells. Every single living cell produces enzymes. It's how life stays alive, is enzymes.
So, nice rule to go by. In biology, if it ends in the letters ASE, it's almost definitely an enzyme. There's very, very few rules other than that. So, if it ends in ASE, it is probably an enzyme. So, for example, catalase, amylase, lipase, phosphorylase, pectinase, these are all enzymes because they end in ASE. If it ends in OSE, it is a sugar. If it ends in AISE, it is mayonnaise, which is none of the above. It is a food substance because it's got that I there. So ASE is an enzyme.
Okay, so enzymes are used to speed up cellular reactions because they're biological catalysts. Without enzymes, most reactions in life would occur too slowly to actually sustain life. Now, this is the reason why humans, for example, are so fussy about temperature, like our body temperature has to be 37 degrees Celsius. That's because that's the best working temperature for most of our enzymes, and that keeps us alive. So the reactions that enzymes catalyze keep them alive and functional. And just a little example of this table is without an enzyme, you're looking at a number of molecules. Say this was a toxin that you accidentally built up in your system. Without the enzyme, that molecule would slowly break down in 4 molecules per minute. With the enzyme, 14,000 per minute. So you can see this idea that if that was a toxin, if that was something that was harmful to you, the enzyme version could get rid of it almost instantly. Without the enzyme, it might take a long time and it might cause some serious harm. Okay, this wee table here is just looking at the idea that all cells produce enzymes. So, for example, there's this enzyme called polymerase. You can find that in plant, animal, bacterial, fungal. It doesn't matter. It doesn't have to be a human, doesn't have to be an animal, but plant, animal, bacterial, and fungal, they all have enzymes inside of them. And sometimes we see the same enzyme across multiple different creatures, species, and that's because it's performing a crucial keeping you alive role.
Okay, so that second key fact, the idea that enzymes speed up reactions, this is kind of a nice metaphor of it in a way with frogs. The idea is if there is no enzyme, basically the reaction is going to happen a lot slower because there's a really, really high energy requirement for the reaction to happen. Whereas enzymes, they can lower the energy required to actually perform the reaction. So as soon as there's an enzyme there, the energy required for that reaction is less, which means it's going to happen more because it's easier to get to a lower energy value than to a higher energy value. So you're going to be able to do things at a faster rate because that energy required to do it is less.
Okay, so enzymes are made by all cells. Enzymes speed up reactions. When an enzyme actually works as a biological catalyst, it remains unchanged in the reaction. So it can be then used again and again and again to do the same reaction over and over. So this is why an enzyme or catalyst is different from stuff in a reaction, like reactants and products, because it's not part of the reaction itself. It's just helping things along, making it easier to happen and speeding that reaction up.
Okay, so two important definitions you need to know. You need to know a substrate. That is the substance on which an enzyme acts, so the thing that an enzyme actually does a job upon. And a product is the thing formed from the chemical reaction. So when the enzyme acts on a substrate, the thing produced at the end of it is the product.
Okay, now key fact number 3, enzymes are specific. The substrate is a specific shape. The enzyme that binds to that substrate is also going to be a specific shape. Now, there are lots of different types of enzymes, therefore there are lots of different types of substrates. Now, the area that the substrate is going to bind to is called the active site. We'll cover that a bit more later. The active site has a complementary shape to the substrate. So if we have a look at this little GIF here, the active site would be this kind of triangular zone here on the yellow Pac-Man, whereas the substrate is that perfectly triangular shape there, okay? It's not the same shape as the substrate, otherwise the active site would be a pokey out triangle. It's a complementary shape. And that means that enzymes are very specific for their substrates. It means that they only act on one particular type. To give you an example, catalase is the name of an enzyme. It only breaks down hydrogen peroxide. It only fits in to hydrogen peroxide. It will not break down any other substance in the world. Only hydrogen peroxide is the right substrate that matches in to the catalase active site.
So how do enzymes actually catalyze reactions? So again, the idea that every enzyme is complementary to a specific substrate. So the substrate and the enzyme actually match. The idea is that when the enzyme and the substrate bind, so when they actually come in together, you can see in that third part of that diagram, it's showing the enzyme and the substrate actually physically binding together. This is when the reaction occurs, is when those two complementary shapes can bind together, the reaction can then occur. And that is the reaction actually occurring and then going on to produce the products.
Okay, now these reactions are quite often drawn like this. Okay, so the substrate is turned into the product. That's where the arrow is going. The enzyme lives above the arrow because it's not involved in the reaction. It is just helping speed things up. Okay, now someone might ask you to write out in this format an actual enzyme reaction. So that means you have to put the name of an actual enzyme above the arrow, the name of its substrate on the left, and the name of the product that is made on the right. Okay, this diagram here is just the general format for an enzyme reaction. So it's a basic template for if you have an enzyme reaction, this is how you write it. You put the name of the enzyme above the arrow, you put the substrate on the left, you put the product on the right.
So here are some common examples of ones that might even come up in exam questions. The top one we've kind of already vaguely discussed, but if you look at them all, they're all written in the same format. On the left-hand side, so our hydrogen peroxide, our starch, and our protein, they are all going to be our substrates. So always written in the left. In the middle, that thing above the arrow in capital letters, so catalase, amylase, pectinase, these are all our enzymes. And then everything on the right of the arrow, they are our products. So in this case, all of these would be read across the way. So hydrogen peroxide, arrow with catalase to water and oxygen. That is one reaction. Starch, arrow with amylase to maltose, that is another reaction. And protein with an arrow with pectinase to amino acids, that is another reaction. But we can identify the substrates from each, the enzymes for each, and the products from each individually.
And you might be asked to do that. You might be given a reaction that looks like this and asked, what is the substrate? What is the enzyme? So you need to know from where is it positioned to pick the right thing. Okay, so I mentioned this before, the active site. Every enzyme has an active site. Okay, now that is the area that is complementary to the shape of the substrate. The active site is a particular region on the enzyme where the substrate is going to bind to. Okay, and it's going to have a specific shape, and it will be the right shape for only its substrate. Okay, different substrates will not fit into the active site because the different enzymes are wrong. So for example, if I was to take the substrate of enzyme 2 and try and shove it with enzyme type 1, that would not work because they do not fit together. Okay, so this is what we mean by enzymes are specific, and the active site is the region on the enzyme where the substrate is going to bind. Okay, so next step, so say the substrate goes into the active site, the next thing that happens is an enzyme substrate complex forms. Now this is where the reaction is going to happen. Now this binding facilitates the reaction. That word facilitates means to make the reaction happen more easily. That is language that you might just need to get used to for the SQA. They might ask you something along the lines of, what process facilitates a reaction when an enzyme and substrate bind? And you have to say they form an enzyme substrate complex. Okay. And what this means is that's just the term that we use for when they're joined together. Okay, so you can see on the left is that substrate and active site and enzyme are separate, and then on the right they have joined together, so we call that the enzyme substrate complex. It's not too difficult of an idea.
So the fourth key fact, the idea that enzymes are involved in either one of two types of reactions. They can either be in a synthesis reaction, we've touched on what synthesis means before, the idea that it builds up, or a degradation reaction, which is something that breaks down. So, yeah, okay. There are your two types of reactions. Yes. So a synthesis reaction looks just like this. It's like the one you've already seen. The idea is it takes multiple substrates, and then when the enzyme substrate complex forms, it produces one or a fewer bigger products. It basically takes lots of small things and produces less big things. And then degradation is the opposite. It takes one big substrate and then it binds and it releases more smaller products. It could be 1 into 2, it could be 1 into 3, 4, 5. It doesn't matter as long as the idea is there are more smaller products.
That link at the bottom is just to a BBC Bite-Size page on enzymes. You might want to peruse it at your own leisure. Okay, so that's a summary of the basics of enzymes. So the idea is an enzyme, and a key term again, flashcards, you want to build this up, is that enzymes are a biological catalyst that speeds up a reaction without being used up.
Okay, the substrate is the substance that an enzyme acts upon.
The product is the substance or substances that are produced at the end of an enzyme reaction, depending on whether it's synthesis or degradation.
Enzyme specificity is the idea that enzymes are specific to one type and one type only of substrate.
The active site is the area on an enzyme that a substrate binds to, complementary to the substrate shape. This is something that people tend to get a bit sticky with, is sometimes they seem to think a substrate does not have an active site. Only the enzyme has an active site.
Okay. The enzyme substrate complex, this is when a substrate binds to the active site of an enzyme and they're joined together, that's that complex.
Okay, an enzyme synthesis reaction is a reaction that builds up multiple substrates into one bigger product.
And a degradation reaction is a reaction that breaks down a substrate into multiple products.
Okay, so that's the basics of enzymes as a start. So the idea of biological catalyst we just covered. Enzymes are produced during protein synthesis in the ribosome. The enzymes are going to speed up a reaction without being used up, so they can get used over and over, and they act on a substrate and they are going to produce products at the end. Very, very, very helpful molecules are enzymes. What we're going to look at in the next video is how the enzymes can be influenced by temperature and pH, and basically what happens if you change those, the effect on enzymes, and then the effect that that can have on a living thing if you change its temperature or its pH.